All right, so welcome everybody to your first remote learning video lecture kind of a thing. You'll have to bear with me. It's a little weird talking to a screen and recording myself and watching myself in the corner. I'm not like a YouTube person or something, so I've never done this before. So yeah, if it's a little awkward, as you like to say, or if it's a little weird, just kind of bear with me and we'll get through it. All right, so anyways, here we go. Hope you're doing well. I'm not sure when you're watching this, but maybe you're having a good morning, good afternoon, good evening. I don't know what you're doing, but hopefully you're here and you're going to learn a little bit about PCR. So by this point, you should have already done the whatchamacallit, you should have done the virtual lab, right? You should have got that taken care of. And now we're here just to kind of go over PCR, and I'm going to try my best to kind of work my way through the slides here and kind of help you understand some of these things. You may know this stuff already, but hey, it doesn't hurt to review it, and it's also kind of like a way for you to look at my basement room. Say, woo, look at it over here, right? So I'm in the office basement. I'm all tucked in down here. Kids are sleeping, so we're actually able to get some work done now. So here we go, and just kind of follow along. And of course, if you have questions, feel free to email, or remember, I'll be available on Google Meet on Monday. That's like the designated meeting time for people to ask questions and things like that, right? So here we go. What is PCR, right? So like we like to say sometimes, PCR stands for polymerase chain reaction. And so it's one of those nice things in science where the name of it tells you pretty much exactly what it is. So you got right here, hey, look at that, right? Polymerase. So hopefully from before spring break and before all this crazy stuff happened, you might remember we were talking about polymerase. What does that do? Remember, it's an enzyme. It's got the ace at the end there, right? So polymerase, what does polymerase do? I'll give you a second to answer that. All right, there's your second. So I hope you said polymerase polymerizes. Remember that polymerase is what copies things, right? It makes polymers out of monomers. So polymerase chain reaction is all about polymerizing. And what are we polymerizing? Well, we're polymerizing DNA, right? So we're trying to make a whole bunch of copies of DNA. And so you might be asking yourself, hey, Self, why in the world would you want to make a whole bunch of DNA? Well, what a great question. The reason why we want to make a whole bunch of DNA is, well, if you are in a lab and you need to do research on DNA, sometimes you only get a little bit of DNA and you actually need a lot. And so PCR will allow you to do that. It will amplify, like you see here. This is an amplifier for people like me who used to listen to guitar music a long time ago. But we're amplifying DNA to create an amount that's useful for experiments, just like it says. So we're trying to make more DNA. So that's part of it. But the main reason, what we're going to really focus on here, is it allows you to locate and copy specific genes. And that's really what is kind of where we're going to take this. That's what we're going to apply this PCR idea to. Specifically, like if you've been wondering how in the world do they test people for COVID-19, for example, and you can use PCR to do that. And a whole bunch of other stuff. We'll get to that later. All right. So I'm already at 3 minutes and 40 seconds. I only got 15 minutes. So we're gonna get going here. So here we go. All right. So what is PCR? It requires all the same stuff that DNA replication requires. It requires a polymerase enzyme, except it's not DNA polymerase. It's something called Taq polymerase. And Taq stands for Thermus aquaticus, which is the scientific name of bacteria that live in these cool little underwater volcanic vents here. And so they like things really, really warm. And so that means that Taq polymerase is able to function at really, really, really high temperatures, which are used in PCR. If you were to put your enzyme in here, your DNA polymerase inside this PCR reaction here, which we'll talk about in a little bit, it would just denature, break down, it wouldn't work at all. So what we need is an enzyme that works at really high temperatures, because PCR uses high temperatures, pretty close to boiling. And if your enzymes boil, they're done for. So we can't use those. We also use primers. Right? If you remember, we used primers in DNA replication. They prime replication and allow it to happen, But more specifically now, what we use primers for is we're going to use primers to find a specific gene that you're trying to amplify and replicate. And so it's pretty interesting. You know what gene you're going after when you do PCR, and you know the DNA sequence. You know what the order of the ATGC is. You know what that is. And so you can design primers that match that sequence, that complement it. And you remember what I mean by complement? The idea that, A matches with T and G matches with C. You got to bear with me. I'm using my mouse because I lost my stylus, so I can't really use it. But anyways, so A with T and G with C. So what you do is you design primers that can locate your gene of interest, because again, you know exactly what gene you're looking for, so you can design primers that attach to it. So, all right, so I made a mistake there. You have to deal with me if there's some weird editing thing. So in either case, again, you design primers to go look for your gene, right? That's basically what they do. So far we've got our polymerase, we've got our primers, and if you recall from DNA replication, what else do we need? Well, we need a whole bunch of nucleotides, right? This slide here just shows you a reminder of what primers do, right? There's your primer there, there's your primer there, and then you end up with a copy in your DNA, right? Here it goes, here it goes. So there you go, you need primers to do replication, right? And then we also need nucleotides, because remember these lines here are nucleotides that keep getting added one by one by one by one, right? So that's how we polymerize. Just like you need bricks to build a building, you need nucleotides to build a polymer, because nucleotides are the monomer. And this walks you through PCR step by step. So remember, in your cells, you have helicase, which unwinds the helix and breaks those bonds and separates the strands. Well, we don't have that in PCR. So what we're doing instead is we're going to denature the enzyme, denature the DNA. So how do we do that? Well, we're going to heat it up at 94 Celsius, which you might know 100 Celsius is boiling. So we're reaching pretty high temperatures here, and that's more than enough to break apart the DNA. So we heat it up and we split apart the double helix. And at this point, remember, we also need the DNA polymerase that can withstand high temperatures. So this is what we're talking about. That's why we have Taq polymerase in there. And so step 2, we lower the temperature a little bit, and then there's something called annealing, which is a fancy word for primers attaching to the DNA, right? You see the primers attaching here and here. So that's the annealing step. And as once the primers are there, then we can start copying. That's called extension over here. So we've got the primer, and then we just go down that way, and then we polymerize our DNA, and we go both ways. Because remember, DNA molecules go in opposite directions, that anti-parallel idea. So we copy our DNA. And so now we've got 2 strands, and then we start it up again, and we go through denaturation, annealing, extension. You basically do these processes over and over and over and over and over and over and over again, about 30 to 40 cycles, depending on what you're trying to do. And by the time you're done, you get an awful lot of DNA, just like it says over here. And PCR is done in a machine here called a thermal cycler. So, thermal, where do you think thermal comes from? I'll stop for you to think about that. Oh yeah, thermal comes from heat. Good answer, right? And then you also get the cycling idea. Well, cycling, of course, you know what a cycle is, right? So we're just going to do this process over and over and over again. So thermal cycler. And so I like this picture because, here's your initial DNA segment. We break apart the black halves of the molecule, and then we copy it, that's in blue. And then we break the black, that goes up here, the blue, that goes here. And then we got purple, and we break them again, and you keep on doing it over and over and over again, and you copy the strand. And so you might remember that each new DNA molecule is half old, half new, right? It's like black is the old, here's the black old one, here's the blue new one, black old, blue old in this case, now purple new, and then here black old, purple old, and this kind of orangish color is the new one, right? So every time we copy DNA, half old, half new, and that old strand is what we call the template. So why in the world would we want to do PCR? Well, we want to do PCR because, again, we're going to copy a bunch of DNA to use for research, but we're going to focus on this idea of finding any gene. So my pictures are a little bit, they hide the text a little bit, but I think you can still see what's going on. So it allows us to screen for genetic diseases. If we know the DNA that codes for a certain genetic disease, we can look for it, right? So if I know the genetic sequence for something like cystic fibrosis, which is a lung disorder, I can screen you to see if you have it. I just have to isolate some DNA from you. I have to add the primers that find the cystic fibrosis gene. And if they find it, then I know you have cystic fibrosis. We can do the same thing to determine parents of children, biological parents, like they used to do on like the Maury Povich show. I don't even know if he's still on, or Jerry Springer or something like that. You can use that to determine biological parents of kids, because each child is going to have a unique DNA sequence that matches their parents. So you run primers based on that. Same thing with who committed a crime. If you find DNA at a crime scene. And what's most relevant right now is this last one right here: screen blood or body fluids for presence of bacterial or viral DNA. So this is literally how they would test to see if you have COVID-19, how you have, if you have anything, like they can determine if you have bacterial or viral pneumonia. They can figure out whatever's in your blood, because if we know the DNA sequence of whatever's in your blood, we can design primers to go find it. And if the primers find it, they're going to spit out this graph that you see right here. We're going to talk more about that later. I might have to make a separate video for that because I'm already at 11 and a half minutes. So I think I might do that because I don't want to rush things. But so PCR is a pretty powerful tool, as you can see. It allows you to do all sorts of things here. So yeah, we'll get to this here, this idea of what qPCR is, because I don't want to rush through that. So basically, if something's made of DNA, or if it has DNA in it, sorry, I'm trying to, no, that didn't work. Whatever. So you can see the slide. Anyways, if something's got DNA inside of it, PCR can find it, as long as you happen to know what that DNA sequence is. That's essentially what PCR is all about, is making a whole bunch of DNA to either do research on that DNA, like in a laboratory, like scientists, quote unquote, who do work on DNA, RNA, cell stuff, or perhaps more, interesting applications would be, all these things here. So genetic diseases, obviously there's DNA involved there. Determining the parents of children, obviously DNA there. Who committed a crime? If you leave blood behind on a crime scene, hair, you're going to leave DNA there as well. So we can figure out who was at a crime scene based on the DNA that's involved. And then same thing with screening body fluids. So if you've got DNA from a virus, DNA from bacteria inside you, we can find it. And there you go. That's essentially PCR. All right. So polymerase chain reaction. Hopefully, again, you've already done the virtual lab. I hope this kind of helped you figure out some stuff. And then your last assignment for the week is to do the flowchart. I know you've done a lot of flowcharts, but when you've got a multi-step process, flowcharts make sense, right? That's a way to kind of review what you know, check your understanding, and see kind of how you're doing. All right. So I'm going to have to post another video about what's called qPCR. Don't worry, it won't be 15 minutes. It's just probably going to be longer than like the 2 minutes I have right now. So I'll post another one about that. And again, if you've got questions, feel free to just reach out and let me know. All right. Thanks for watching. And I've watched enough YouTube videos. I got to say, if you like what you saw, please give a thumbs up, subscribe, enable notifications, or support us on Patreon, right? Do all that good stuff. Don't actually do that stuff, but I just thought I had to say it. All right. Hope you're doing well, and we'll see you soon. All right. Take care. Bye-bye.